Okay, now we're talking about the ribosomes. The ribosomes. The ribosomes are little granular looking objects that have a protein coat, like a marble, with some ribosomal RNA inside. A protein coat with some ribosomal RNA inside. What is the job of the ribosome? It acts as a construction table to make protein. So its total job is to act in protein synthesis, the making of proteins. A very vital role. The ribosomes are made in the cell nucleolus from the previous. That cell nucleolus, who is in the nucleus, has the genetic material and the bits and pieces of ribosomes. Okay? We have two groups of ribosomes. They're just alike, but we call them free ribosomes and fixed. Free and fixed, or some will call them membrane-bound ribosomes. Free ribosomes and fixed. Here's something to study. If a protein is made on a free ribosome, that protein is to be used for some purpose inside the cell that made it. If a protein is made on a free ribosome, it is to be used by the cell that made it. If the protein is made on a membrane-bound ribosome, also called a fixed ribosome, then it is to be used either in the cell membrane or exported from the cell. Exocytosis. So proteins made on membrane-bound ribosomes will be used either in the cell membrane or outside the cell. Okay? I'll skip that picture. Ribosomes are particles made of ribosome RNA and a protein coat. Ribosomes carry out protein in two locations. In the cytoplasm, they're floating around to be used. The product protein is to be used inside the cell. Or fixed. Now, if it's fixed, it's going to be fixed on the endoplasmic reticulum, which is the organelle we're going to talk about immediately, right after I finish this ribosome. Okay? So free ribosomes, protein used inside cell, fixed, used in the cell membrane or outside the cell. Now, ribosome is made in the nucleolus in two components, a small subunit and a large. Listen to me, a small subunit and a large. So when the ribosome exits through the nuclear pore, see, it's being made in the nucleolus, then it exits into the nucleoplasm. Nucleolus and then exits through the nuclear pore. Let's go back to an earlier drawing. So the ribosome would be made in the nucleolus, then travel to the nucleoplasm, then it would exit through these holes. When it exits through the holes, though, it exits at a small subunit separate from a large subunit. So they exit separate. They exit separate. They exit separate. But when they get ready to be called forth in the cell cytoplasm for a job, then the two subunits will click together and form this. So they exit initially, and until they're ready to be used, there'll be a separate large subunit floating in the cell cytoplasm and a small subunit floating in the cell cytoplasm. All right? So review. Ribosome used construction table protein synthesis. Ribosome made in nucleolus. Ribosome exits nucleolus as a small subunit and large subunit and stays separate until the job is called forth. And when the job is called forth, that's when it occurs. Now, if something is made on a floating ribosome, the protein product is used inside the cell. If it's made on a fixed ribosome, fixed to the endoplasmic reticulum, the product is used either in the cell membrane are exported from the cell, exocytosis. So now we go to this endoplasmic reticulum. Let's break the word down. Inside the cytoplasm reticulum. Reticulose in Latin means thready. So this is a thready looking substance inside the cytoplasm. Okay? The endoplasmic reticulum accounts for more than half of the total membrane in a eukaryotic cell. The ER is continuous with the nuclear envelope. There are two distinct regions. Now regions, not two ERs, but two. So it's one long thread. One long thread. Part is what we call the smooth ER, and the other is the rough. The smooth ER lacks ribosomes, and they never will sit there. The rough ER has ribosomes on its surface. Now I want to discuss this real quick. In the old days, the rough ER could not be distinguished as the rough ER unless it had the ribosomes. Let's move to modern day. You can reserve a table at a restaurant, not be there, and they know that's where your table is. So the rough ER, even without the ribosomes, has receptors. Those receptors are where the ribosomes would sit. So even if the ribosomes are not there, a cytologist, cell person, could say that this is designed for the rough ER. The smooth ER does not have the receptors, so no matter where the ribosomes are, they cannot affix. All right? So here's an example. This is smooth. This is rough. In this case, you see in the ribosomes. You can even see a difference in the shape of the rough versus the smooth. Rough versus the smooth. This still is one long string, basically. The smooth ER are tubules that are arranged in a looping pattern, you can see. The main thing about this is the proteins, because this is an endoplasmic reticulum membrane, the proteins in here can act as enzymes in lipid metabolism. So in other words, synthesis of lipids can happen in the smooth ER. It's also a big area for breaking down material in your body. Chemicals, whether taken from the outside or inside, particularly in the liver. In the liver, that's where a lot of breakdown of pesticides, carcinogens, all your chemicals. Synthesis of steroids. Skeletal muscle is a very important area for storing calcium, we'll talk about, known as the sarcoplasmic reticulum. So the sarcoplasmic reticulum is another name for the smooth endoplasmic reticulum in muscle. Okay. That's the smooth. What about the rough? The rough's job is really totally involved in protein synthesis. Now, it can be involved in protein synthesis and hooking carbs to it. But its job, the rough ER, is really dedicated to dealing with protein synthesis. Okay. Here's the smooth. Here's the rough. Here's the smooth. Just to show you some pictures. So the endoplasmic reticulum surrounds the nucleus. It's contiguous with the nucleus, and then it kind of takes a tortuous pattern. I don't have a good picture. A tortuous pattern throughout the cytoplasm, and the other end of this tube, let's say, is located close to the cell membrane. It has two regions, but one long string. The smooth ER and the rough ER. They do different things, as we alluded to. They do different things. We go next to the Golgi. The Golgi, from a person's name, F.M. Golgi, the Golgi consists of flattened membranous sacs called cisternae, box of chest. The Golgi can modify, package, sort, and secrete, let's say, material that comes from the endoplasmic reticulum. So what the Golgi gets comes from the endoplasmic reticulum, the rough ER. What the Golgi gets comes from the rough ER. Whatever it gets, it can modify, package, sort, and secrete that. So it makes these transport vesicles. So here we are. The Golgi are these flat sacs. You have different groups. It's like stacked together, these plates. The plates closest to the endoplasmic reticulum are called the cysts. Those in the middle are called the mesh, and those on the outside are called the trans. The trans are where vesicles occur that will move material to the cell membrane for export or to be placed in the cell membrane. So anything coming off the rough ER must pass through the Golgi. Anything coming off the rough ER must pass through the Golgi. Coming off the rough ER would be proteins, glycoproteins, and such. They would then enter into the cis plate of the Golgi, be modified, packaged, secreted, and then modified, packaged, and then export from the trans to go to the cell membrane for either placement in the cell membrane or export. So here's the rough ER. You pinch a vesicle off the rough ER. This is actually called the transitional ER. There'd be a vesicle coming. The vesicle then traveling from there, inside the vesicle would be glycoproteins or new proteins, would then go into and hook to the cis plate of the Golgi. The material then would go from here and then go to the next plate, and it would be modified, maybe modified materials, maybe cut off, so forth. And then it would keep going, and then that would pinch off, go to the next one. That would pinch off until finally you get to the trans. The trans then will pinch off and give this secretory vesicle coming from the Golgi. That vesicle then would come and fuse with the cell membrane and either be used in the cell membrane as a component or secreted. So anything coming off the rough ER is going to go to the Golgi. Now remember, proteins made on the rough ER would be used either in the cell membrane or secreted for the outside of the cell. Exocytosis. So we'll close here. Our next organelle will be the lysosomes, which actually comes from the Golgi. All right.